So for the final project, we're going to be talking about advances in medical technology. So one of the things that I was really interested in researching more was wireless brain sensors. So through medical advances, doctors and scientists have been able to work together and create electronics that can be placed in the brain and can aid doctors in monitoring temperatures, pressures, and take other measurements within the brain. Dr. Thomas explains these sensors important, saying these are important to help understanding how the brain works and to communicate with it, as in brain-computer prosthesis. The brain-computer interface can be used either to analyze or to produce such neural electrical signals in order to diagnose or treat neuropsychiatric conditions. So, what are some of the applications of wireless brain sensors? They can be used in cognitive assessments. They can help monitor brain injury. They can enhance neural function. They can be used in pain management. They can be used to monitor and help epilepsy. And they can be used in sleep assessment. So the next thing I want to talk about is nanotechnology in medicine. There are many different ways in which nanotechnology is currently being used and developed in medicine. For example, researchers at Worcester Polytechnic Institute are using antibodies attached to carbon nanotubes in chips to detect cancer cells in the bloodstream. The researchers believe this method could be used in simple lab tests that could provide early detection of cancer cells in the bloodstream. So what are some other types of nanotechnology in development? Well, there's drug delivery, there's diagnostic techniques, and that's what the cancer diagnostic quote was about. There are antibacterial treatments, there's wound treatments, and there's cell repair. So now on to my favorite new development in the medical field is CRISPR-Cas9. CRISPR is a new innovation in gene editing. CRISPR-Cas9 is one of the most precise types of gene editing to date. It edits genes by precisely cutting DNA and then letting DNA's natural repair process to fix the cut. So a couple things that we can do with CRISPR. We can develop new kinds of drugs, we can alter someone's genome to eliminate a disease, and we can treat blindness. There are so many things. We can treat blindness, we can treat HIV, we can treat cancer. It is just such a cool technology. So how CRISPR works. I had to do a little bit of research into this for another class, and that's why I'm so excited about it. So the Cas9 protein um, binds with a guide RNA in a cell. And so this complex attachment matches a DNA sequence to a spacer. And so then the guide RNA leads the Cas9 to where it needs to be. So the Cas9 cuts the double strands of the DNA, and then the programmed DNA can be inserted into the cut. And by doing that, you can implement new types of DNA. You don't have to do a pre-programmed slice of DNA. You can just edit out a section of DNA and then the DNA will just naturally heal itself. And then, so these are my sources, and I hope you enjoyed. Thank you.